Let's talk a little bit about facilitated diffusion. In facilitated diffusion, that's where a carrier molecule moves substances into or out of cells from areas of high concentration to areas of low concentration. This, like diffusion and osmosis, is a passive process. What I mean by passive process is that no energy is required to be expended by the cell for this to occur. The number of molecules transported is directly proportional to the concentration difference. So once again, a concentration gradient is needed for facilitated diffusion to occur. In facilitated diffusion, there's a protein in the membrane of the cell, and this protein changes shape in such a way to regulate the amount of passage of a certain molecule. The example here on the right of the slide is showing a sugar molecule or a glucose molecule being transported across a cell wall, one glucose molecule at a time. Now, the only way that this will happen is if the concentration on this side is much lower than the concentration on this side. So there's far more sugar molecules on the right side of the cell membrane and far fewer on the left. If there were the same concentration on each side, even facilitated diffusion would not move the substance across the cell membrane. Active transport is another mechanism by which materials are moved across the cell membrane. In active transport, a substance is moved across a concentration gradient, or even against against a concentration gradient. So what's meant by that is that a solute is moved from an area of low concentration to an area of high concentration, or exactly opposite of the way that natural diffusion occurs. Active transport requires energy as well as some type of a carrier mechanism. Active transport mechanism may exchange one substance for another. The best example of active transport is the sodium-potassium pump. The cells in your body use the sodium-potassium pump, in particular muscle cells and some, skeletal muscle and cardiac muscle. And the sodium-potassium pump, generally speaking, there's a higher concentration of sodium outside of the cell and a higher concentration of potassium inside the cell. The cell exchanges, I think it's 3 sodium molecules for 2 potassium against the concentration gradient to maintain the difference in concentrations. By doing this, a electrical charge is produced on the outside of the cell versus the inside. This allows for the cell to be polarized inside to outside and allows for gates to be opened that allow the ions to rush back, or the sodium ions to rush back into the cell, and allows the cell to depolarize. This is how muscle cells contract, by the way. We'll talk about that more in the next unit. Filtration is another process by which materials are moved across the cell membrane. Filtration does not require any energy, so it's a passive process, and it does not require a concentration gradient. Instead, it requires a pressure gradient. There must be a difference in hydrostatic pressure on one side of the membrane than the other. If there's enough pressure, hydrostatic pressure, then the material will move across the membrane. The force that pulls the water back into the capillary from the interstitial space is called onconic force, or capillary action. No one really understands how that works exactly. It's just been observed for quite some time to exist. An example of onconic force is, you may be familiar with, if you take the thin paper wrapper from a drinking straw and you place it on a little bubble of water on a table, you'll notice that the water is pulled up into the fibers of the paper of the drinking straw wrapper. That's onconic force, or capillary action. Endocytosis is an active process which requires energy. It literally means the eating of material by a cell. So it's the uptake of material through the cell membrane by a membrane-bound droplet or vesicle that forms within the protoplasm of a cell. Endocytosis has 2 types. There's phagocytosis, which literally means cell eating. This is when endocytosis takes up solid material. Penocytosis literally means cell drinking, and this is when the endocytotic action takes up liquid. Exocytosis is the reverse. That's when the release of secretions from the cells is the most common example of exocytosis. The secretions accumulate within vesicles within the protoplasm. The vesicles bind to the inside of the cell membrane, and then the contents of the vesicles are eliminated from the cell. Examples included secretion of digestive enzymes by the pancreas, mucus secretion by the salivary glands is another example.